Hello, wonderful AP Bio students, and welcome to our video on cell cycle control. In your body, you have 100 trillion cells, and on average, daily, 100 billion of them divide. That's about 1/1000th of all of your cells. The reason that it's on average is because some cells don't divide at all, and some divide very, very rapidly. For example, your neurons, they don't divide at all. Your liver cells only divide if there's damage to the liver, so that the liver can be regenerated. And your muscle cells don't divide at all. Even when they're harmed, they cannot divide to repair themselves. However, your intestinal cells divide very, very rapidly through the process of digestion. So let's look at the stages of the cell cycle and how your cells themselves control those stages. Here is a common diagram that you will see often of the cell cycle, and it shows 3 different stages, G1, S, and G2, and all 3 of these are part of interphase. I crossed that out. You should know that. This is all part of interphase. And then when the cell's not in interphase, it's undergoing mitosis, which is, of course, the division of the cell into 2 cells, actively dividing cell. So let's talk about each one of these stages and how they are controlled. The first stage I'd like to talk about is G1. And G1 stands for growth 1. And basically, I want you to think of this stage as like going to Home Depot. You have one house, you want to build another house, so you need to get all of the necessary raw materials to build that house. And that's exactly what the cell is doing in G1. It's preparing for division, it's duplicating the organelles, it's getting everything ready in terms of the raw materials to become 2 cells. Now it's doing everything, it's duplicating everything except for the DNA. So in growth 1, everything is duplicated. Next phase is the S phase. Let's do a different color for that. And S stands for synthesis. And specifically, when I talk about synthesis, I'm talking about D N to the A, DNA synthesis. This is when each of the 46 chromosomes, I'm going to abbreviate chromosomes like this, each of the 46 chromosomes is copied through the process of DNA replication. So this is when your cell is making an exact copy of the genetic information found within the nucleus. So remember, the cells that we're talking about here are diploid cells, so they have 2 sets of chromosomes. And eventually we're going to be making 2 diploid cells. So we're starting with 1 diploid and we're ending with 2 diploid, and all of these cells have a perfect exact copy, hopefully, of the chromosomes in the original parent cell. So synthesis phase is when DNA is copied. Our next phase is G2 phase, and that as well stands for growth, but it's growth 2. And during this phase, the cell continues to grow, it gets larger, and also an important thing that happens is the double checking of DNA to make sure that there is no errors. And this is when the cell is preparing for division, because the next phase we're looking at is mitosis. And mitosis, of course, is none other than PMAT-C, which stands for prophase, metaphase, anaphase, telophase, and cytokinesis. So this is when active division is occurring and we end up with 2 diploid cells. The process in which the cells actually physically divide is cytokinesis, and that's when we get that little, it's called a cleavage furrow, where it's kind of indenting on itself and it pinches off, and now we have 2 separate cells. In plant cells, it forms what's called a cell plate, and then the cells separate from each other. So there we have the cycle. Now it seems simple enough, but there's a lot of things that can go wrong. And because there's a lot of things that go wrong, our cells have what's called checkpoints put in place. Now I like to think of checkpoints as the dial or the washing cycle on your washing machine. So when your washing machine goes, you put your clothes in, it fills up with water. Once it's filled up to a specific point, which would be a checkpoint, then the little thing inside of it starts to turn, right? And once it's been washed good enough, it drains and then it spins. And so there's specific stages and specific triggers for your clothes in your washing machine to be able to move on to the next cycle. For example, how the little spinny thing inside can't start spinning unless it's filled with water. It couldn't spin dry clothes. So that's kind of what our cell does. It has checkpoints. So starting at G1, your cell's moving along this nice little path, and it enters what's called the G1 checkpoint. And this is kind of like, I think of it as like when you're driving on the freeway and they have those toll booths and you need like money to get through. So the G1 checkpoint, it's basically receiving a go-ahead signal in order to move to the next phase of the cycle. And these actually use signal transduction pathways in order for the cell to move into the next section of the cycle. So basically what we're looking for in these checkpoints is if crucial cell processes have occurred and they have completed correctly. G1 checkpoint is pretty well known for animal cells, and it's often called the restriction point. And if the cell can get past this restriction point, if everything in G1 has occurred correctly, then typically the cell will move through G1, S, and G2, and eventually dividing in the M phase. Sometimes though it doesn't, and it says, hey cell, you're not ready to divide yet, so I want you to enter G0. And G0 is kind of like the inactive, in between, sedated point in which a cell is at rest. And a lot of your cells are at this stage right now, specifically the cells that don't divide at all. Like your neurons are always at G0. They're not going to be actively dividing. So these checkpoints put in place are really important to know if things have been done correctly and are actually done. Another thing that controls cell growth is two things. The first thing I would like to talk about is anchorage dependence. Anchorage dependence. And that basically means a cell has to be attached to something in order to divide. And we can see here, this is a little petri dish. We see here that some cells have attached themselves to the petri dish. The second thing that is worth talking about is called density dependent inhibition. And sometimes this is also called contact inhibition. And it's basically that, like, when cells divide, they kind of know when it's getting crowded and they stop dividing, so they're not overcrowding the area. So that's where density comes from. So certain things will trigger the cells to stop dividing if there's not enough resources or space to divide. And if you take this petri dish, so you can see they have this nice even layer, and they're crowded, but they're not going to overcrowd it. If you remove a few cells, they'll actually go in and fill that space because some space has become available and they've been able to divide and fill it. So those are two kind of things that are kind of an overarching control of the rate at which cells divide. Now let's get a little deeper and look at the molecular control of cell division. Two things I want to introduce you to. One is protein kinase, which you know a lot about already. So remember, protein kinase is basically just a protein that is used to phosphorylate other proteins to activate them. So protein kinase is a friend that you need to be familiar with. And also another protein found in your cells called cyclin. Let's talk about the two of these in our next slide. So cyclin right here is a fluctuating protein. So it fluctuates in concentration, fluctuates in concentration, there we go. It fluctuates in concentration depending on what phase a cell is in. And this cyclin is actually kind of an activator for kinase. And this right here, other puzzle piece here, CDK stands for cyclin dependent kinase. So this is a kinase, remember, a phosphorylated protein that is dependent on the presence of cyclin in order for it to act. So the activity rises and falls with the concentration of cyclin. These two love each other. When the two of them get together, they form what's called, let me find another color, here we go. They form what's called MPF. And MPF stands for maturation promoting factor. Or the way I think of it as M phase promoting factor, because it basically triggers a cell to go from G2 to M phase, where the cell is actively dividing. So now that we know the players in this beautiful cycle here, let's talk about the cycle itself. So step 1 in the cycle, in late S phase, right about here, cyclin is developed. So synthesis of cyclin is developed in late S phase. So here we have an increased presence of S phase, and of cyclin. And cyclin is protected in this part of the cell cycle from degrading, so it accumulates. Let me put a step 1. Cyclin accumulates. So in step 2, let me change colors here. How about a red? In step 2, the accumulated cyclin combines with CDK, cyclin-dependent kinase, which is kind of abundant in the cell. It's floating around in the cell, producing MPF. So when the two of these are together, they produce MPF. So step 2, accumulated cyclin, so CDK binds with cyclin, and that produces MPF, which is the M phase promoting factor. So the high presence of MPF is basically the ticket to pass through the G2 checkpoint. So if you are driving on a toll road and it's $2.75 to keep going, they're not going to let you through unless you have $2.75. If you have $2.60, they're not going to let you through. So the presence, a specific amount of MPF present is kind of the trigger to get through the G2 checkpoint and initiate mitosis. So in step 3 here, we'll do it in blue. In step 3, we have mitosis. This is, step 3. So MPF promotes mitosis, and the way it actually promotes it is since it's a kinase, it phosphorylates other proteins which are involved in actively dividing the cell, involved in mitosis. So after mitosis occurs, we have our step 4. I'm going to have to go back to a different color. Which one did I say? Oh, we've got black. We have step 4 right here, where basically conditions in the cell degrade cyclin. So cyclin is degraded. And then the CDK is recycled and kind of waiting for more cyclin to be produced. So there are our stages. Let's go through it one more time. So in late S phase, cyclin accumulates. And when cyclin is abundant, it bonds with CDK, cyclin-dependent kinase. And the two of these form MPF. And we need a certain amount of MPF to basically pass through the G2 checkpoint and actively cellular divide. And once the cell is divided, the cyclin is then degraded and it's kind of recycled and used to be built up again later in the S phase. And the CDK kind of waits around for cyclin to build up a bit. I kind of think of cyclin and CDK as a tumultuous relationship. So cyclin, or sorry, CDK is like the one that's always there. It's waiting around. It's like the good one. And cyclin, or sorry, yes, CDK is always there. And cyclin is like the crazy one that's like going up and down and up and down. And when they're together, they're good. They're that awesome, good MPF molecule. But sometimes that doesn't always last. That's my analogy. It's a bit of a stretch, but it works. So let's look at this within the cell and how the levels of both activities connect with each other. So these little arrows right here, these signify the stages of mitosis. So let's look here. We have G1, we have S, we have G2, and we have mitosis. And if we look at this line right here, this one that kind of has this sharp peak and then a drop off, this is going to be our cyclin concentration. So cyclin concentration. And this little hump right here, let's make it blue, is going to be our MPF activity. Wow, that's a really bad tracing. So this is our MPF activity. So we can see that through our cell cycle, once we get to the M phase, so through S and G1, our cyclin concentration is increasing. And once we get to the M phase, we've made our MPF, right? So we mitotically divide, and then our cyclin drops off drastically because it can't survive in the cell at that point. And the whole thing goes through as it goes through the different phases of mitosis. So, in review, we talked about cell cycle in the body, which cells go through it often and which cells don't. The cell cycle phases, which include G1, S, G2, and M. Oops, there we go. And we also looked at cell cycle control. We looked at the specific checkpoints. There's 3 checkpoints within the cell cycle. And we also took a look at CDKs, which is cyclin-dependent kinases, and how it binds with cyclin to make MPF, which essentially pushes the cell from G2 into the M phase. And that is our video. Thank you for joining me.